The aim of this procedure is to visualize messenger RNA that is associated with the endoplasmic reticulum, or ER. This is accomplished by first plating mammalian tissue culture cells on cover slips. The second step of the procedure is to treat the cells with extraction buffer to remove cytoplasmic mRNA not bound to the ER. The next steps are to rapidly fix the cells and stain the mRNA by fluorescent in situ hybridization. The final steps are to image and then quantify the amount of fluorescence associated with the ER. Ultimately, results can show how mRNAs are associated with the surface of this organelle under different conditions.
This method can help answer key cell biological questions, such as whether mRNAs that encode secretory proteins can be maintained on the surface of the ER independently of ribosomes or protein synthesis.
Visual demonstration of this method is critical, as it involves a few tricky cellular manipulation steps.
Seed the tissue culture cells on acid-treated cover slips for at least 1 day prior to the experiment. COS7 and U2OS cells are both suitable choices as they have high secreted protein production and well-defined ERs. If an exogenous mRNA is being investigated, transfect the cells 1 day after seeding and then incubate the cells for an additional 18 hours to allow for mRNA expression. On the day of the experiment, to achieve high extraction efficiency, the cells should not exceed 80% confluency on the cover slip. To detect